Hello, my name is Ribka Erdiana, and today I will do science communication regarding the definition, working principles, and application of PCR to my friend Dylan.
Hi, I'm Dylan. I live in the United States, so because of the great distance between us, we might have some technical issues that we have to sort out, so I apologize for that. My level of education is high school graduate with some college, about 2 years of college. Um, that's about it.
What do you know about polymerase chain reaction or PCR?
Next to nothing. We've talked briefly about it before, but not being a scientist or a biologist myself, I don't really remember a whole lot. I know it has to do with the replication of DNA, I think, right?
Yep, you're correct.
Okay.
So, polymerase chain reaction or PCR is a method of amplifying DNA. So, we take a small amount of DNA and amplify it to a large amount. Amplifying here means like to copy and copy it again. Do you know what a DNA or genome is?
I know DNA is deoxyribonucleic acid.
Mm-hmm.
Um, might have to refresh my memory on what a genome is. I mean, I know the Human Genome Project. It's well, let me guess. A genome would be the sequencing of an organism's DNA, right? Or
Mm,
or is that
you're
not it?
close. You're close.
Close. Okay.
So the genome is like the whole sum of the DNA. For example, the genome of a cell, that means that the whole sum of the DNA in that cell. And so that means a human genome means the whole sum of the DNA in that human. So a genome is like the instruction manual that makes you, and the DNA is like the language it's written in. So if the
Okay.
genome is, yeah.
Okay.
So
Gotcha.
in relation to PCR, if the genome is the whole instruction manual and DNA is the language it's written in, then PCR is a method for searching and copying a specific sentence or paragraph in that instruction manual. So because DNA is very small and oftentimes in the samples we've got, very few in amount, the goal is to make enough copy of the specific sentence or paragraph you want so that it's readable for further analysis. That sentence or
Okay.
paragraph could be a gene researchers want to understand, or a genetic marker used by forensic scientists to match crime scenes with DNA with suspect. Is that okay so far?
So I think so. So basically, when you do PCR, it would be like if you have a small piece of DNA.
Mm-hmm.
Are you essentially creating copies of that to allow yourself to do multiple tests when you otherwise wouldn't have enough material to do multiple tests?
Yes, that's one of the application.
Okay. Yeah, so I wouldn't know how we would even create DNA out of nothing like that because that, like I said, I don't really know anything about this. So that would be my first question. I guess you're the one doing the interview, but the first
Mm-hmm.
question that comes to mind for me is like, how are we just creating copies like that?
Okay, that's actually my next question as well. So, how do you think PCR works? But if you still have no idea, then I guess I can explain it.
Yeah, I don't know. I would want to know what the new genetic, like, what's what's the copy made out of? I
Oh,
would want to know that.
so
But you can stay on script if that's going off script.
no, that's okay. Like, uh, to answer your question briefly, the copy is also made of DNA. So we use an enzyme that can make more DNA, but for the process, to start from the beginning, PCR uses the present DNA in a sample as a template to generate more of a specific region in that DNA. And to do that, it cycles through several different temperatures repeatedly. Is okay, the DNA is original double-stranded, right? Deoxyribonucleic acid is double-stranded.
Mm-hmm.
And first, we need to separate the strands. And we use heat to do that. Like at a certain temperature, usually 50 at 95 degrees Celsius, the double-stranded DNA separates. This step is called DNA denaturation. So this is the first step of PCR.
Okay, I do remember that from our previous conversations,
Mhm. So after the
and
DNA
I
denature,
am taking notes here, so that's why I keep looking down.
oh, that's fine. That's fine.
Okay, so denaturation is using heat to separate the helix, basically, right?
Yep. hmm
Okay,
Okay. And we do this so
I'm
that
with you so
primers.
far.
hmm And we do this so that primers can attach to the separated strand. Primers come in pairs, forward and reverse, each for each strand of DNA. Primers are like a looking glass, like using Control F on a digital book. Primers look for a
Now you're
specific
speaking my
region.
language.
Yeah. So yeah, I hope I'm using the metaphors correctly. So primers look for a specific region in the DNA, a specific sentence or paragraph in the instruction manual, and then attach to it. When the temperature is lower, it allows the primer to attach to the now single-stranded DNA. We call this step annealing, to anneal the primers to the single-stranded DNA. Okay, the next step is DNA synthesis. So aside from the specificity, primers are important because they give the DNA starting point for DNA synthesis. We call this step the DNA extension. The DNA synthesis is carried out with the help of an enzyme called DNA polymerase. DNA polymerase. So from the name itself, you can guess that it functions to make more polymers of DNA. To, facil- to facilitate the step, we raise the temperature to around 70 degrees Celsius. In the reaction mix, you have DNA, the sample, primers, DNA polymerase, and a bunch of nucleotides. So DNA polymerase doesn't create nucleotides, but instead match them to the DNA strands because the nucleotides are already in the reaction mix. So the DNA polymerase is just attaching them to the matching the DNA strand.
I thought I thought that's what happened during the last step, though. I thought during the last step, during annealing, that the primer attached itself to the similar DNA
Yeah.
strands.
So the this is the primer, and then the primer has an open end. Primers are this is the DNA strand, for example, and this is the primer. The primer has an open end where the DNA polymerase can attach and start inserting nucleotides that match the DNA strand. That's why the primers are important. They have an open end, for example.
Okay, so the primer almost seems like it's an anchor point for the polymerase
Yep,
to
yep.
connect to. So like
Mhm.
you have the DNA, and then you have the polymer. Sorry, no, the primer. You have the primer up here, and then it seems like the polymerase would get like connected along there. It's just kind of like it's like two pieces of a jigsaw puzzle, and it just fits itself. Okay,
The primer has an open end, the polymer attach, and then went along the DNA strand attaching nucleotides.
okay. So I think I get it.
In a typical PCR, the cycle repeats about 25 to 35 times, which usually take 1.5 to 2 hours, depending on the length of the DNA region being copied. So the longer the region you're copying, the longer it takes usually.
I
And
figured. Yeah,
mhm,
that makes
and the
sense.
target region can go from just one copy to millions or billions of copy in that time range. That's because it's not
Right.
the
So
original DNA that's used as a template each time. In fact, the new DNA that's made in one round can serve as a template in the next round of DNA synthesis. Thus, the number of DNA molecules can roughly double every cycle.
okay. All right. Well, was that the whole process? Did we cover everything?
Yep, that is the whole process.
Oh, okay.
Uh, Do you think you understand about PCR at
Yes.
least?
Yeah,
Can you
I actually do. Can I explain PCR? Okay, so PCR, you start with a strand of DNA, a sample. You denature it, which means you're heating it up to split the helix into two sections, and then you're taking a primer, um, putting that on there, which I guess automatically kind of seeks out, like the. Hold on, I wrote it down. What's the? Thought I wrote it down. Nucleotides. The nucleotides automatically kind of like seek each other out from what it sounds like, and then that kind of serves as like an anchor for the polymerase that you then put on top. So it's almost anchored on like two sides. You got like the primer on one side, and then the uh, originate strand on the other, and then it just kind of fits right in there like a puzzle piece. And then you have with the primer and the polymerase, you have a full copy of the original side of that DNA strand.
Mm-hmm.
And then you can keep doing that as many times as you want, I guess, as long as you have the primer and the polymerase. But yeah, that sounds like that's the gist of it anyway. And I am happy to say I think I understand it better. So thank you for explaining that to me.
And thank you for listening.
My
Can you
pleasure.
think of an example of PCR, by the way?
An example? You mean like an application?
Mm-hmm.
Well, we already discussed the forensics.
Actually, one of the most well-known applications of PCR that well-known recently is in COVID-19 screening. If you've heard of the
Oh,
PCR test.
I did actually. Yeah, I never I didn't make that connection.
Mm-hmm. Yeah, it also uses PCR to viral genetic material because in the patient, especially a asymptomatic patient, the viral genetic material is just so low that you
Yeah,
need to amplify it first to be able to detect it.
that's really cool. That's cool though.
Mm-hmm. And thank you for listening and just wanting to learn about this.
Oh yeah, no, I mean this stuff is interesting. I like learning new things in an informal setting.